Okay, everyone. So for today's presentation, we're going to try to cover photosynthesis and the pentose phosphate pathway. There might be people who join in the presentation in real time. So if there are people who do that, I will try to answer the questions. But um, other than that, if you have any questions, feel free to email me as usual. And for those who are watching this in real time, feel free to send a message over chat, and I will read those and also answer any kind of confusion. So that being said, to cover the photosynthesis and the pentose phosphate pathway topics that hopefully you've watched Render Hide and Explain, I kind of want to preface it by saying that photosynthesis is basically the reverse of the cellular respiration you've learned so far. You've learned how we go from sugar to the TCA cycle to break it down into smaller bits, and then we use all of that to garner energy and use an electron transfer chain. Now we're going to do the opposite. We're going to do an electron transfer chain and build molecules so that we can generate basically a biomolecule such as a sugar. And so to start off, the first phase of the photosynthesis pathway is the light-dependent reactions. And it's more because light powers the movement of electrons and generation of energy. So to start off here, I have a diagram of kind of what it looks like from a simplified view. Essentially, light will come in and it does two things. It splits water so that it would release electrons. And in the process, you create your first product, oxygen. Additionally, not only does it split the water, it also indirectly powers the pump of protons. So when you think about protons being pumped into a direction in photosynthesis, that is driven by light. And hopefully you guys remember that photosynthesis occurs in what we call the chloroplast. And generally, the structure of a chloroplast is this kind of big membrane, kind of like the mitochondria, but there's another membrane inside that is kind of like pancake shaped. And so to give you a definition of the parts, the space inside is called the stroma. The pancakes itself are called the thylakoids. And then what you call the inside of the thylakoids, you can call it the lumen of the thylakoid, or you can call it the thylakoid space. But essentially, it's what's inside the thylakoid. And so in general, when you do photosynthesis, the direction that we want to pump protons to is the inside of the thylakoid. And that's because it's the most compact space. And if you have the most compact space, then you can generate a proton gradient faster. In the mitochondria, if you guys remember, there's an intermembrane space. The intermembrane space is generally smaller, and that's why we pump protons there. So that's kind of the analogy between the mitochondria and the chloroplast. And then the proton gradient is used to form ATP. And so if you were to remember something by heart, it should be this page that you don't need a cheat sheet on. You should remember light powers the splitting of water to produce NADPH and ATP. How exactly that happens, we'll go over on the next slide, but make sure you have this in mind. So before we start, I want to talk about how light enters this whole pathway. And I know there's some physics involved because he started drawing some like electron diagrams. So I'm going to try to draw it as simplified as I can. And so light is involved in these pigments like chlorophyll that we've heard of. Essentially, they are metal ions that are conjunction with a lot of aromatic rings. And so it's kind of like a heme that we've learned so far, except in this case, it's a solar panel. This is the silicon if you would think of plants as a solar panel. And then so whenever you think of these electron acceptors or carriers, what we're referring to initially are these pigments. So hopefully you can imagine that what this diagram is referring to is this metal ion inside. So hopefully you can find that connection. So when light hits a pigment molecule or a metal, you hopefully remember from Introchem that what can happen to it is raise an electron to an excited state. And we can see here that you have one electron at the lower ground state and one electron in the higher state. And so this is an energetically rich molecule then. And so there's two ways this can be returned back to normal. Either the electron just comes back down, and if it comes back down, usually it releases light or heat. And so usually that's what we learned in introchemistry. In biology, there's a special way to get rid of this excited energy. And actually that's to just give it to another carrier. And so if we like to think of this first molecule, the excited molecule D, as a chlorophyll B, for example, and then maybe this is a carrier next to it, then one way the chlorophyll B can get rid of the excited electrons is to transfer it to its nearby carrier. And in the process, we now have a acceptor with an extra electron, which is why it's negative. And we have now a initial pigment that has no electrons, missing electrons, and so that's why it's positive. And so in photosynthesis, we're going to show a lot of diagrams where electrons might be hopping from one thing to another. In that scenario, what's going on is this. If we show you that, actually, I'll show that later in another diagram. But if an electron moves from one place to another, what's going on is this diagram right here, where an excited electron is actually directly transferred to another carrier. The other way that electrons and energy kind of move about is to kind of demonstrate here. So in the earlier example, we had an electron transfer. In this scenario, I'm going to show you, you don't necessarily actually need to transfer an electron itself to make another carrier excited. You can just transfer the energy. And so this is what they call resonance energy transfer, if you're ever interested. But for the sake of this exam, you don't even need to know how to name this. But basically what's going on in this kind of energy transfer is, let's say light hits an electron at the first photosystem pigment. And so in this case, you hit the location with the light, and so you get an excited electron. After you get an excited electron, you don't necessarily have to transfer the electron itself to the next pigment. And let's say you don't want to generate ions. So instead, what you can do is make this electron come down, and in the process, the energy released from that can be used to activate or excite the electron from the nearby pigment. And so in this scenario, what we've done as a whole is transfer this excited energy from one pigment to another without having to transfer any electrons. And so when I show you later these diagrams where you have one pigment to another, kind of like pointing like this, what we're trying to say here is this molecule gets excited by light, and then this molecule then transfers that energy to the next one, and transfers the energy to the next one. And when you think about what that's going on, then you should think about this step right here, where no electrons are actually being moved, but energy is being transferred as one electron goes back down. On the other hand, in the center of the photosystem, which we'll talk about later, I'll draw a different diagram where instead I'll show you that an electron is actually moved from one place to another. And in that scenario, that would be like this, where an actual electron moves. And so the final product are ions. Any questions so far for the people who are here in real time? Okay, so now that I've talked about that as a background, let's actually talk about where this applies in photosynthesis. So I put in here the pathway in the most concise way and how as much as you need to know, in my opinion. So essentially, there are 4 parts, just like how there's multiple complexes for the electron transport chain. There are a couple complexes for photosynthesis. You don't need to memorize the flow of electrons on the inside, but what's important for you to know is the products formed, and you can also just write down your cheat sheet what the pathway is inside. And so how do we generate NADPH, ATP, and oxygen is how we want to think about this. So to first start off, light hits photosystem 2, and it splits water, and that's how we get the oxygen. So essentially what happens here is the light will power the release of electrons from water, so that the electrons from the water can now enter the electron transfer chain. So the electrons that flow on this chain come from the water. And then in the same time, the H plus is now pumped into the thylakoid space. And so this process of pumping protons and moving electrons from water is an energy needing process, and that is why we need light. And the carrier that takes the electrons from there is Q. And you guys have remembered Q from last time. Q is a carrier, and then the reduced form is QH2. So in this case, Q picks up electrons and becomes QH2. And then after that process, QH2 will give its electrons to another carrier. So in this diagram, what this is showing is that QH2 gives the electrons and becomes Q again. And in the process, what takes the electrons is this carrier that we shorthand for PC, or plasticyanine. And basically, we have an oxidized form and a reduced form. And so that will take the electrons. And in the process, protons are popped. If you are thinking this is kind of the same repetitive process, one thing gets the electrons and then gives it to another thing, it's exactly what's going on. But throughout the process, we're pumping protons. And so if I were to kind of follow the pathway of an electron, the electrons start with water, then they move and become part of QH2. And then QH2 then gives it to this enzyme complex, which gives it to PC. So then it goes to PC. PC will give it to now this new complex in the middle, which has this molecule called ferredoxin. So it gives it to ferredoxin. And then ferredoxin will then give it to NADPH at the end. And so as a result, we basically have a net movement of electrons from water to NADPH. And then the proton pumping occurs kind of like in the process. Essentially, in these 2 complexes, protons are pumped, but at the same time, protons are kind of being consumed because we need protons to build NADP positive. But also, when you split water, H plus also leaves into the stroma. And so then you guys already know when H plus builds up in the center, then that can flow through ATP synthase to form ATP. So what you should know by heart, you should know what the products are by heart. You should know at least the general geometry of this, but you can write down your cheat sheet kind of like where the electrons moves from each step. And know that light is involved in 2 areas here. It is involved in photosystem 2 and photosystem 1. And why? So the first part, light was important because it splits the water and does the first pumping of electrons. Great. The second part is actually very crucial. If you guys remember, NADP to NADPH is an energy requiring process. In order to create this energy storage in the form of an electron carrier, you need to have a high energy electron. But at that point, usually here there's not enough energy in that electron. And so what we're trying to do here is have light come in and charge that electron again so that we can put it into a carrier and we can use this for reducing purposes later on. And so we need to reduce things later on. So we need to have a very high energy electron. And so that's why we want to use light again. So that's the purpose of this second light, is to re-excite and re-give energy into our electrons. Any questions? Okay, let me clear my drawings. So to give you then, I know I talked about this earlier, but I wanted to just show you what that looks like from a theoretical perspective in a chloroplast. So if we zoom in into a chloroplast, into a thylakoid, a photosystem kind of looks like a giant cylinder protein. And if we zoom that in even more, you get this whole complex right here. And so on the side of a photosystem, so this whole thing is the photosystem, you have a bunch of pigments. And then in the center, you have what's called the reaction center. And so what happens is light hits the pigments on the side. And when that gets excited, they excite their neighbor. And in the similar process we talked about here earlier. Then they excite their neighbor, then they excite their neighbor, and then eventually you'll reach to the reaction center, where a special reaction happens. When the reaction center gets excited, instead of exciting its neighbor by releasing energy, it instead gives up its electrons to a carrier. And so if you guys remember, the carrier for photosystem 1 is Q. And so when this reaction center gets excited, instead of transferring its energy to another thing, it will instead give it to Q so that it becomes QH2. And this is also where the reaction of splitting water happens, because once you remove the electron from this reaction center, you create a very strong oxidizing agent. And that is where water comes in, because water needs to replenish the electrons that was lost in that reaction center. So that's what it looks like geometrically in a chloroplast. Do you need to be able to explain this geometry in the exam? Not really, but it helps to understand sometimes why this occurs, so that you can remember how it works. All right. I don't have enough time to go over the cyclic versus non-cyclic, but I put information here just in case. But essentially what you should remember, in my opinion, besides maybe a diagram of this, is the difference in the products. The cyclic pathway of photosynthesis, which doesn't use water, if I can summarize it, does not use water. This one only produces ATP, which makes sense. Water was important because it gives us the electrons. So if you don't use water, you don't get the electrons. On the other hand, non-cyclic, which uses water, in this case, because we use water, we have an electron source. So hence we produce NADPH and ATP. So that's one thing to remember. I like to remember it that way. But light is involved in both, so that's why I know ATP will be made. But water is only involved in one of them, and so you should not produce NADPH on one of the pathways. Feel free to look at these notes in the meantime, but I want to go over the more complicated stuff. So hopefully this is also, remember this from intro bio. Okay. Moving on to the next part now, which is the Krebs cycle. It's the TCA version for the plants, except it's the reverse. We're making sugar instead of breaking it down. And so hopefully a lot of these pathways will have mechanisms you've seen before. And so hopefully you can recognize the enzyme. The enzymes that function here, except for some unique ones, will be pretty much the same. So hopefully that mechanism you wrote down in your cheat sheet in the past, you keep. So let's talk about this. So usually when we talk about the cycle, we had one thing coming in and one thing that was like the precursor that it stuck to. So for the Krebs cycle, the input is the CO2. So if I were to create a TCA cycle analogy, this will be the acetyl-CoA. This is the thing that enters the pathway. What accepts it is this molecule called ribulose 1,5-bisphosphate. And using the enzyme Ribisco, they get combined together to form a 6-carbon intermediate, which then splits into 2 3-carbon intermediates. And so the way it enters the cycle is exactly the same way it does for the TCA cycle. You have one molecule that kind of cycles, and you have your input, and they combine together. And then in the process, without going too specific, because I'm just giving you a general description, once this two combine, the way we're going to generate a sugar for glycolysis or a sugar to make sugar, and also to recycle is using the ATP and ADPH form. And so hopefully the way you should think about this, we take CO2, the most oxidized form of sugar or of carbon. And we're going to take that from this step and turn it into a sugar, which is much more reduced. And so, and higher energy. And so this process needs electrons and it needs energy. And so as a result, that is where the light-dependent reactions come in. They provide these two. And like the TCA cycle, you're left with, you have to both produce something, but also you also have to replenish the ribulose 1,5-bisphosphate we made. So all of these concepts I talked about hopefully resonate with the TCA cycle. But I don't really think you should memorize the specific compound names if you have in your cheat sheet. But the concepts like that, you should have them in mind so that you can be aware of what's going on. it's less likely for a vendor hiding to ask you a mechanism pathway from photosynthesis connecting